Welcome back to the deep dive. It is great to have you with us. Today, we are looking at an organ that, honestly, is a bit of a paradox. It is absolutely essential for the existence of the human species. I mean, none of us would be here without it. Yet it is the only organ in the human body that is designed to be disposable.
Right.
We use it, and then we literally throw it away.
It's often disregarded as just medical waste, or, in the delivery room, it's the afterbirth.
The afterbirth.
The thing that comes out after the main event. It's an afterthought.
Exactly. But looking at the stack of research you've pulled together today, specifically synthesizing the placenta and umbilical cord chapters from I.B. Singh, and then the clinical depth from Moore and Prasad's The Developing Human, and of course Langman, I'm realizing we have this completely backwards.
Oh, we do.
This isn't just a wrapper. This thing is a biological masterpiece.
It really is. It is a shame it gets such a bad rap because if you look at the physiology, the placenta is arguably the most complex, multitasking organ the human body ever produces. And here is the hook that usually gets people, the thing that sets it apart from your heart or your liver. It is the only organ formed by two different individuals working in concert.
That's the two worlds part of our title today.
Precisely. It is not just the mother, and it is not just the fetus. It is a collaboration.
No negotiation.
It's a physiological handshake between two distinct biological entities.
And it's doing everything. That's what jumped out at me from the Moore and Prasad readings. For 9 months, this temporary organ is acting as the fetus's lungs,
The kidneys,
liver, digestive system, and immune system, all at the same time.
all while managing a very, very complex negotiation between two organisms that, under any other circumstances, might actually try to reject or kill each other.
Which is a terrifying thought we will definitely get to in the immunology section. So our mission today is to unpack that negotiation. We're going to look at the architecture of this lifeline, the incredible fluid dynamics involved, the bloodbath that feeds the baby,
And the hormonal takeover that happens in pregnancy. And just a quick note on the sources before we dive in. We are synthesizing very dense chapters here. I.B. Singh is fantastic for the structural organization, those exam-oriented facts, while Moore and Prasad and Langman give us that rich clinical context.
Right. And as always, we are here to spark curiosity. If you are a med student listening to this on the way to a rotation or prepping for exams, go look at the diagrams. Seriously, you cannot understand the placenta fully without seeing like figure 9.5 in Singh or the clinical plates in Langman. We are here to give you the story behind those diagrams so that when you look at them, they make sense.
And the theme of that story, if I had to boil it down, is that survival depends on relationship. Biological growth cannot happen in isolation. It requires a dialogue.
So let's unpack this dialogue. Let's start with the architecture. You called it a handshake between two worlds. Who are the partners in this handshake?
Well, you have two sides coming together to build this bridge. On the maternal side, you have the uterus, specifically a layer we call the decidua bassalis.
Okay, I want to pause on that word decidua. I looked up the etymology on this because it sounded familiar. It comes from the Latin for falling off or shedding.
Exactly.
Like deciduous trees in autumn losing their leaves.
That's precisely it. It's named that because, unlike the rest of the uterus, which stays put after birth, this functional layer is destined to be shed. It serves its purpose, and then it leaves. But before that happens, it acts as the soil. This is what we call the decidual reaction.
This was interesting to me. It's not just passive tissue waiting for a seed. The text says these cells undergo a massive transformation.
Oh, drastically. It's a preparation for a siege. Driven by high levels of progesterone, these endometrial cells, they enlarge, they get massive. And they fill up with glycogen and lipids, basically storing fat and sugar.
So they're stockpiling.
They are. Under a microscope, they look pale because they're so stuffed with nutrients.
So they're getting ready for something.
They are prepping for a famine. You have to remember, when the embryo first arrives, there's no umbilical cord, no blood vessels connected yet. These decidual cells act as a pantry, stockpiling energy, to feed the early embryo via diffusion before the plumbing is fully connected.
I see.
They also play a crucial defensive role, protecting the mother's deeper tissues from being uncontrollably invaded by the embryo.
Okay, so that's the mother side, the prepared, nutrient-rich soil. Who is the other partner?
The fetal component. And this is the chorion frondosum.
Chorion frondosum. That sounds like a spell from a fantasy novel. I cast chorion frondosum.
It does, doesn't it? But the Latin is actually quite descriptive.
Yeah.
Frondosum refers to fronds, like fern leaves. It means leafy or bushy.
Ah.
This is the part of the fetal sac that develops these massive branching projections called villi.
So if we stick with the gardening metaphor, the mother provides the soil, the decidua basalis, and the fetus puts down roots, the corium frondosum.
That is a perfect metaphor. And just like roots seek water, the fetal trophoblast, those outer cells of the embryo, they invade the uterine wall seeking blood.
Okay.
Now, to visualize this, you have to understand the geography. When the embryo burrows into the uterus, it's not just sitting on the surface. It's like a seed planted deep in the earth. The uterus encapsulates it completely.
The texts mention 3 different deciduas based on where they are relative to the baby. Help us visualize that, because it can get confusing on a 2D diagram.
Okay, let's try. Imagine the embryo is a tennis ball buried in a thick foam mattress.
Okay.
The part of the mattress directly underneath the ball, forming the base, that's the decidua basalis. That's the foundation. That's where the placenta formed.
We're at a base.
Now, the part of the mattress that has been pulled over the top of the ball, covering it like a blanket, that's the decidua capsularis. It encapsulates the fetus.
Right, the capsule.
And then the rest of the mattress surface, far away from the ball, lining the rest of the room. That's the decidua parietalis.
Like the wallpaper of the room.
Exactly. And here's the cool dynamic part. As the baby grows, that capsule pushes out. The tennis ball becomes a basketball, then a beach ball. Eventually, the capsule pushes all the way across the uterine cavity until it touches the wallpaper on the other side.
And they fuse.
They fuse and the capsule disappears.
Yeah.
But the main action, the handshake we are talking about, is all happening at the base, the decidua basalis.
So let's look at those roots, the villi. I.B. Singh breaks this down into stages: primary, secondary, tertiary. Is this just academic trivia for the exam, or does it matter for understanding the function?
Oh, it absolutely matters.
Yeah.
It's the story of how you build a life support system from scratch.
Okay.
A primary villus is just a solid column of cells. Think of it as a plastic tent peg. It provides structure, it holds the place, but it's not alive inside. It has no core.
Okay, so just a structural anchor.
Then the secondary villus forms when the mesoderm, that's the connective tissue layer, pushes into the center of that peg. Now the peg has some bulk. It's got a core of tissue, but still no blood.
And tertiary.
That's when the magic happens. Blood vessels form inside that mesodermal core. Now, it's not just a peg, it's a pipe. It is capable of transport. And by the end of the 3rd week, this vascular network is established.
So we go from a solid peg to a vascularized root system in just 3 weeks. That is incredibly fast construction.
It has to be. The embryo is growing exponentially. It needs resources now.
Right.
And these villi aren't all doing the same thing. We differentiate between anchoring villi and floating villi.
The names seem pretty descriptive.
They are. The anchoring villi are structural supports.
Yeah.
They attach the placenta firmly to the uterine wall, forming what we call a cytotrophoblastic shell.
Okay.
They are the nails keeping the structure in place so the placenta doesn't just slide off the uterine wall.
And the floating ones?
Those are the functional units. They branch off the sides of the anchoring villi, and they dangle freely in the space between the mother and the fetus. They're like seaweed floating in the ocean currents, just maximizing their surface area to soak up nutrients.
Which brings us to part 2 of our outline, and honestly the most fascinating part of the anatomy for me, the ocean they're floating in. This is the intervillus space.
This is where the human placenta is quite unique and where it breaks the standard rules of anatomy.
How so?
You know, in most tissues in your body, blood stays inside blood vessels. If blood leaves a vessel, that's usually a hemorrhage. It's a bruise or it's internal bleeding. It's bad news.
Right. We generally want to keep the blood in the pipes.
But the placenta is what we call hemochorial. In this organ, the maternal blood actually leaves the mother's vessels.
It's a literal bloodbath.
Ideally, we say it bathes the villi, which sounds a little more relaxing. But yes, the spiral arteries from the mother punch through the floor of the placenta and just spew blood into this open space, the intervillus space.
The numbers here are wild to me. I.B. Singh says this space holds about 150 milliliters of maternal blood. That doesn't sound like a huge amount, like half a soda can.
It's not a large volume at rest, but the key is the turnover rate. That 150 milliliters is replenished 3 to 4 times per minute.
Wait, let me process that. That means the entire pool is replaced every 15 or 20 seconds.
Correct. It's a high-flow, high-pressure system. Those spiral arteries act like jets. They squirt blood under high pressure toward the roof of the placenta, the chorionic plate.
Like a fountain.
Exactly like a fountain. The blood shoots up, hits the top, dissipates pressure, and then it trickles down over those floating villi, washing over them so they can extract oxygen before draining out through the endometrial veins at the bottom.
So the fetus is essentially dipping its roots into a constantly refreshing river of its mother's blood.
Without ever actually mixing the blood. That's the key. The mother's blood is in the open space.
Mm-hmm.
The baby's blood is inside the vessels, inside the villi. They're close, microscopically close, but distinct.
I want to get to the mixing or the prevention of it in a moment, but visually, if I'm holding a placenta after birth, which thankfully I am not right now, but if I were, the two sides look totally different, don't they?
Completely different. It's a telltale sign of that two-world origin. If you look at the side that was attached to the mother, it looks rough, fleshy, dark maroon. We call it a cobblestone appearance.
And the cobblestones are the lobes, the cotyledons.
Right, called cotyledons. There are about 15 to 30 of them, separated by little walls called septa. That's the raw, earthy, maternal side that was ripped away from the uterine wall.
And the baby's side.
It looks like something from a clean room. Smooth, shiny, gray. It looks futuristic. That's because it's covered by the amnion, the membrane that holds the fluid.
Mm-hmm.
And right in the center, usually you see the umbilical cord planting itself like a tree trunk with big vessels radiating out under that shiny surface.
It's really quite beautiful in a clinical way.
It is.
Okay, so we have the architecture, we have the pool of blood, but you said the blood doesn't mix. This brings us to part 3, the placental barrier. Although reading more in Prasad, he seems to take issue with the word barrier.
He does, and rightly so. Barrier implies a wall that nothing gets through, like the Great Wall of China. Moore prefers placental membrane or even a sieve.
A gatekeeper.
A gatekeeper, exactly.
Mm-hmm.
It lets the good stuff in, keeps most of the bad stuff out, but it is definitely not impermeable.
And this wall changes over time.
It has to. Think about the needs of the fetus. In early pregnancy, the priority is protection. The embryo is fragile. So the membrane is relatively thick, 4 distinct layers of cells and tissue.
A brick wall.
A brick wall. You have the syncytiotrophoblast, the cytotrophoblast, the connective tissue, and the endothelium. It's secure. But as the fetus gets bigger, its demand for oxygen skyrockets. And oxygen moves by diffusion. It just drifts across.
Right.
You can't have a thick wall slowing down oxygen transfer when you have a 7-pound baby trying to breathe.
The wall comes down.
It thins out.
Yeah.
By the 4th month, the connective tissue and the cytotrophoblast layer disappear in many places. We're left with just a paper-thin layer, the syncytiotrophoblast, and the endothelial wall of the vessel. It goes from a brick wall to a screen door.
Maximizing the flow.
Maximizing diffusion. The distance between mom's blood and baby's blood becomes incredibly small to allow for rapid gas exchange.
Let's talk about how things actually get across this screen door, because it's not just drifting, is it? The placenta is doing work.
It's doing a lot of work. There are 3 main ways things cross, and understanding them explains a lot about fetal health. First, as we mentioned, is simple diffusion.
Okay.
This is for gases. Oxygen and carbon dioxide. They just flow from high concentration to low concentration.
Right.
The placenta is acting as the fetus's lungs here.
Then there is active transport. This is the one that caught my eye because it implies the placenta is working against the gradient.
Right. The fetus is in a rapid construction phase. It needs building blocks, amino acids, calcium, vitamins, and especially glucose. Often the fetus needs these at higher concentrations than the mother has in her own blood.
So if it relied on diffusion, the stuff would flow the wrong way. It would flow back to mom.
Exactly. If it were just diffusion, nutrients might flow back to the mom to reach equilibrium. So the placenta has to pump them uphill. It uses energy to grab these molecules from the mom's blood and shove them over to the baby.
That's hoarding resources.
It acts like a very aggressive shopper.
Yes. And the third one, cell drinking.
Pinocytosis. This is fascinating. This is for massive molecules that are too big to fit through the pores or pumps. The placental cells essentially engulf a droplet of maternal plasma, form a bubble around it, and ferry it across to the other side.
And this is how antibodies get across.
Specifically IgG antibodies. This is critical. This is how the mother passes her library of immunity to the baby. If the mom has had measles or the vaccine, she passes those antibodies to the fetus via pinocytosis.
So the baby is born with the mother's shield.
We call it passive immunity. It protects the baby for the first few months of life while its own immune system is booting up. It's a parting gift from the mother.
But the text notes a gap here. It's not a perfect shield.
No. For some reason, antibodies for pertussis whooping cough and chickenpox, which is varicella, do not cross as effectively.
Which explains why whooping cough is so terrifying in newborns. They don't have that borrowed armor.
Exactly. It's a vulnerability in the system, which is why we emphasize vaccination so strongly for those around the baby.
So the good stuff gets in. What about the bad stuff? The barrier fails sometimes.
It does. We know that many viruses, rubella, Zika, cytomegalovirus, can cross. Drugs like alcohol cross very easily. We call these teratogens. But there's another leak that is clinically very important. Sometimes fetal red blood cells escape the other way into the mom.
This sets up the Rh factor issue, doesn't it?
It does. This is a classic immunological clash.
Walk us through that, because it's a standard exam question, but also a real clinical issue.
Okay, so say the mother is Rh negative. She doesn't have the Rh protein on her blood cells. But the baby is Rh positive, inheriting that from the father.
Right.
If the placenta is perfect, it's fine. But if there's a micro tear during delivery or trauma, a few fetal cells leak into the mom's blood.
And her immune system sees them.
And it screams, intruder. She makes antibodies against the Rh protein. Now, usually, this doesn't hurt the first baby because by the time she makes enough antibodies, the baby is born.
But the memory remains.
Right. Her immune system remembers. For the next pregnancy, those antibodies are ready. And because they are IgG, they can cross the placenta via that pinocytosis we talked about. They go in and attack the second baby's blood, destroying its red blood cells.
Causing anemia and potentially heart failure in the fetus.
Exactly. That's why we have the RhoGAM shot now. We give it to Rh negative moms to prevent that sensitization from ever happening. We basically hide the fetal cells from the mother's immune system before she can react to them.
That leads perfectly into part 4, the immune paradox. You touched on this. The mother's immune system identifying an intruder. But wait, the fetus is half dead. Genetically, it's distinct.
It is. It has foreign DNA.
In any other context, like if I gave you my kidney, your immune system would recognize it as not self and destroy it immediately unless you took heavy drugs. Why doesn't the mother reject the fetus? It's a giant foreign object growing inside her.
This is one of the greatest mysteries in immunology. We call the placenta an allograft, a transplant from a different individual. But it survives.
Is it just hiding?
Partly. The outer layer of the placenta, that syncytiotrophoblast, is in stealth mode. It lacks the major histocompatibility antigens, the MHCs.
The ID badges.
Right. Every cell in your body wears an MHC badge that says, I belong to host name. The immune system patrols, checks badges, and moves on. The placenta takes this badge off, so the mother's immune surveillance flies by and essentially sees nothing. It's invisible.
But it's not just passive hiding, is it? The text implies it's active.
Oh, it's chemical warfare. The placenta isn't just hiding, it's suppressing the local police force. It secretes immunosuppressors locally, progesterone, prostaglandin E2. And there's this specific enzyme mentioned in the research called IDO.
IDO, what does it do?
Indolamine 2-3 dioxygenase. It breaks down tryptophan.
Tryptophan, like in turkey.
Yes, an amino acid. But here's the strategic brilliance. T cells, the immune system's attack dogs, need tryptophan to survive. By breaking it down locally, the placenta essentially starves the T cells that get too close. It creates a demilitarized zone around the baby.
That is incredible. I'm not just hiding, I'm starving anyone who comes to look for me.
Survival at all costs. The placenta is creating a safe haven.
But this invasion, it has to happen just right. What happens if the handshake goes wrong? The texts mention pre-eclampsia.
This is a major clinical topic. Pre-eclampsia is a condition where the mother develops dangerously high blood pressure, but Moore describes the root cause as a failure of this placental invasion.
The failure of the roots.
Yes. Remember we said the fetal cells invade the uterus and remodel the spiral arteries to make them high-flow fountains.
Right. They widen the pikes.
In pre-eclampsia, that remodeling fails. The fetal cells don't invade deep enough. The arteries stay narrow and tight. The placenta doesn't get enough blood.
So you have a starving placenta.
And a starving placenta is a dangerous thing. It starts releasing toxic signals into the mother's blood proteins that damage the mother's own blood vessels. It's basically screaming, I need more flow.
So it hijacks the mother's system.
It creates a systemic crisis. These signals cause the mother's blood vessels to constrict all over her body, spiking her blood pressure to try and force blood to the placenta.
Wow.
But that high blood pressure damages the mother's kidneys, liver, and brain.
So the mother's illness is actually a symptom of the placenta fighting for survival.
Exactly. It's a desperate attempt by the organ to get fed.
Right.
And that is why the only cure for preeclampsia is delivery of the placenta. Once the organ is out, the signals stop, the screaming stops, and the mother recovers.
That is chilling and fascinating. It really reframes the condition. Okay, let's move from the placenta itself to the conduit, part 5, the umbilical cord.
The lifeline.
First off, anatomy. It's not just a rubber tube. It's filled with this stuff called Wharton's jelly.
Which is my favorite anatomical name. Wharton's jelly is a mucoid connective tissue. Think of it as a natural airbag or a firm gelatin.
What is it for?
Its job is to prevent the vessels inside from kinking. The baby is doing gymnastics in there, flips, kicks. If the cord were just loose vessels like headphone wires, they would knot and cut off the blood supply instantly.
And that would be game over.
Immediately. The jelly keeps the cord stiff enough to resist kinking, but flexible enough to bend. It's an engineering solution to a mechanical problem.
Now let's look inside the jelly. The vessel count is counterintuitive for anyone who remembers high school biology.
It trips up medical students all the time. There are 2 arteries and 1 vein.
Usually, arteries carry oxygenated blood to the body, and veins carry dirty blood back to the heart.
But here, you have to think from the perspective of the fetal heart. The umbilical arteries are carrying blood away from the fetus to the placenta.
So that blood is
Deoxygenated. It's full of waste and CO2. It's going to the placenta to get scrubbed.
and the umbilical vein.
It's carrying blood from the placenta to the fetus. So the vein is carrying the clean, oxygen-rich blood. It's just like the pulmonary loop in an adult, where the pulmonary vein has the oxygen.
Got it. So the vein is the supply line. The arteries are the exhaust pipes. The text also mentions that there used to be 2 veins.
In early development, yes, a right and a left, but the right one disappears. We are left with only the left umbilical vein. Why nature decided one was enough, we don't fully know, but it works.
Here's where it gets really weird. The physiological hernia. Between weeks 6 and 10, the cord acts as a backpack.
Chuckles. That's a good way to put it. At that stage, the baby's liver and kidneys are growing so explosively that the abdominal cavity is literally too small to hold the intestines. There's no room at the end.
So what happens?
The intestines loop out of the body, through the belly button, and hang out inside the umbilical cord.
So the baby's guts are hanging out in the cord.
For about 4 weeks.
Oh.
It's a normal part of development. Then, as the body catches up in size, the intestines rotate and slide back inside.
And if they don't,
That's a birth defect called an omphalocele. But normally, it's a temporary relocation. It shows just how dynamic this process is. Nothing is static.
let's talk about knots. The cord is long, the baby is moving. Do true knots happen?
They do, but they are surprisingly rare, about 1% of births. Thanks to Wharton's jelly, even if a knot forms, it usually doesn't pull tight enough to stop the flow. But a more dangerous condition is something called velamentous insertion.
That sounds ominous.
It is. Normally, the cord plugs right into the thick, fleshy center of the placenta. It's a sturdy connection. But in velamentous insertion, the cord misses the mark. It attaches to the thin membranes on the side, away from the main placenta.
So the vessels have to travel to get there.
Yes, the vessels have to run across the delicate membrane-like wires running across the floor without a cover to reach the placental tissue.
And they aren't protected by Wharton's jelly there.
Exactly. They are exposed. If the membranes rupture during labor, when the water breaks, those exposed vessels can snap. It leads to massive fetal blood loss in seconds. It's rare, but it's something doctors look for on ultrasound because it changes how you have to manage the delivery.
Let's zoom out a bit. The baby isn't just floating in a vacuum, it's floating in water. This is part 6, the waters of life, the amnion and amniotic fluid.
The spacesuit and the ocean inside it. The amnion starts as a tiny blister over the embryo and expands until it obliterates everything else, creating a sealed, fluid-filled bag.
What is the fluid actually doing besides making a mess on the floor in movies when it breaks?
It's a multi-tool. First, it's a shock absorber. It protects the fetus from physical trauma. If the mom bumps into a table, the fluid disperses the force.
Makes sense.
It's also a thermostat, keeping the temperature constant. But crucially, and this surprises people, it's a lung trainer.
A lung trainer. But the baby doesn't breathe there.
No, but it practices breathing. The fetus makes breathing movements, inhaling amniotic fluid into its lungs.
Breathing water.
It sounds wrong, but it's vital. The hydrostatic pressure of that fluid expands the lung tissue. It pushes the alveoli open. If there's no fluid, the lungs don't grow. They stay hypoplastic, underdeveloped.
So if the water breaks too early, the lungs might not work at birth?
Precisely. The fluid is structurally necessary for lung growth.
And the baby swallows it.
It's a recycling system. The fetus swallows the amniotic fluid, absorbs it into its blood, filters it through its kidneys, and then urinates it back out into the amniotic sac.
So, I have to ask, the baby is swimming in its own pee.
Chuckles. Sterile pee. But yes, in late pregnancy, the volume of fluid, about a liter, is largely determined by fetal urine output.
Which explains the pathologies mentioned in the text, polyhydramnios and oligohydramnia.
Right. It's a plumbing logic puzzle.
Yeah.
If there's too much fluid, polyhydramnios, it often means the baby isn't taking it away, so maybe the baby isn't swallowing. Is there a blockage in the esophagus?
And if there's too little fluid, oligohydramnios.
It implies the baby isn't adding to the pool. Maybe the baby isn't peeing. Did the kidneys fail to develop what we call renal agenesis? So measuring the fluid is a proxy for checking the baby's internal organs.
And if there's too little fluid, that leads to Potter sequence.
Yes. This is a mechanical consequence. Without the water cushion, the uterine wall presses tight against the fetus. It squashes the baby. The face gets compressed, flattened nose, low ears. The feet get clubbed because there's no room to move. And most critically, as we said, the lungs don't develop.
It really highlights how the environment shapes the anatomy.
Absolutely.
The text also has some scary images of amniotic band syndrome.
That's when the amnion ruptures inside the uterus, but the chorion stays intact. The amnion forms these sticky, fibrous bands that float in the fluid. If the baby gets a hand or a foot tangled in one, it acts like a tourniquet. It can actually amputate a limb in utero. It shows just how delicate these membranes are.
Before we move to the hormones, I want to touch on the yolk sac. In birds, that's the food, but in humans,
It's a vestigial hero. It doesn't have yolk. We get nutrients from the blood. But early on, the yolk sac functions as the first blood cell factory, what we call hematopoiesis. And surprisingly, it's the source of the primordial germ cells.
wait, the cells that will become the baby's sperm or eggs come from the yolk sac.
Yes, they originate there, and then they migrate into the embryo to find the ovaries or testes. So in a way, the genetic future of your grandchildren starts in the yolk sac.
That is wild. It's like a seed bank. Okay, part 7, the endocrine powerhouse. The placenta isn't just a filter, it's a gland.
It takes over the hormonal control of the pregnancy. The ovaries do the work at the start, but the placenta eventually says, I got this.
The first one we all know is HCG.
Human chorionic gonadotropin, that's the I'm pregnant signal. It's what turns the stick blue. Its job is to tell the ovary, don't shed the lining, we have a passenger, keep the progesterone coming.
But then there is HPL, human placental lactogen. The text links this to gestational diabetes. This felt like a conflict of interest between mom and baby.
It is a fascinating example of evolutionary tension. HPL modifies the mother's metabolism. It makes the mother's cells slightly resistant to insulin.
Why would it do that?
Well, insulin helps your cells absorb sugar. If the mother is resistant to insulin, her body doesn't absorb as much sugar from her blood. That leaves more sugar circulating in the blood.
Which then crosses the placenta to the baby.
Exactly. The placenta is intentionally making the mom a little bit diabetic to make sure the baby gets the first cut of the sugar supply.
Talk about a demanding house guest.
The most demanding. Usually the mother's pancreas just works harder to overcome this resistance. But if her pancreas can't keep up, she develops clinical gestational diabetes. It's the result of the placenta pushing too hard.
Eventually, this organ ages. It has a lifespan.
It does. It's a temporary organ. Towards the end of 40 weeks, it starts to calcify. We see syncytial knots, bits of the placenta breaking off and floating into the mother's circulation.
Where do they go?
They lodge in the capillaries of the mother's lungs, where she dissolves them. It's a sign the organ is finishing its job. It's degrading.
And then parturition, the exit.
The afterbirth.
Yeah.
Once the baby is born, the uterus contracts hard. This shears the placenta off the wall. But here is a critical clinical point. The doctor must inspect the placenta after it comes out to make sure it's complete.
Because if a piece is left behind,
If a cotyledon is missing, it prevents the uterus from clamping down fully. That leads to hemorrhage. It's a life-threatening emergency.
and sometimes it doesn't want to come out at all, placenta accreta.
This is becoming more common. This is when the roots go too deep. Instead of stopping at the decidua, they grow into the muscle of the uterus, the myometrium.
Like tree roots cracking a foundation.
Yes, it won't separate. Often this requires a hysterectomy to stop the bleeding. We see it more now because of C-section scars. The placenta loves to latch onto that scar tissue.
It's a powerful, aggressive organ.
It has to be.
So, bringing this all together for the outro, we started by calling it medical waste. But we've seen it's a lung, a kidney, a bodyguard, a hormone factory, and a negotiator.
It's a masterpiece of adaptation. It serves its purpose perfectly, and then, gracefully, it leaves. Without it, the species wouldn't exist. We are all here because a placenta fought for us.
But does it really leave? This is the final thought I wanted to bring up. The outline mentions microchimerism.
This is where the science gets really poetic.
Yeah.
We mentioned those fetal cells leaking into the mother. For a long time, we thought the mother's immune system cleaned them all up after birth.
But they don't.
Recent research shows they don't always. We have found fetal cells male DNA in women who had sons living in the mother's brain, her heart, her skin, decades after the baby was born.
So the mother is carrying a literal piece of her child forever.
And not just hanging out. These cells appear to be active. They sometimes migrate to sites of injury, like a healed C-section scar or a damaged heart, and try to differentiate into tissue to help repair the mother.
That gives a whole new meaning to the lifeline between two worlds. The dialogue doesn't end at birth.
It never really ends. You leave a part of yourself behind, and you take a part of your mother with you.
On that mind-bending note, we are going to wrap up. I highly encourage you to open the textbooks, look at the diagrams of the villi in I.B. Singh, look at the clinical images in Moore and Langman. It makes this all very real.
Absolutely. The text is the map, but this story is the territory.
Thanks for diving deep with us. We'll see you on the next one.